Welcome back. In this video, we're going to talk about how the dopamine hypothesis changed over time. So remember that the dopamine hypothesis 1.0 stated that schizophrenia results from excessive dopaminergic activity, specifically in the mesolimbic pathway. However, due to some interesting findings, then the dopamine hypothesis has changed. So in the 2.0 version, they changed the definition to that schizophrenia results from a combination of subcortical dopaminergic hyperactivity and cortical dopamine hypoactivity. So too much dopamine in the subcortical pathways and too little activity in the cortical pathways. So the revisions to the dopamine hypothesis was really because many studies conflate psychosis with schizophrenia. So psychosis is certainly associated with dopamine dysregulation, but it's actually only one feature of schizophrenia. So dopamine dysregulation and psychosis represent the cumulative result of genetic and environmental changes to the neurobiology and neurochemistry. Pretty much just saying that other neurotransmitters are also involved in circuit changes upstream to the mesolimbic dopamine. So just examining dopamine doesn't explain everything. So now current antipsychotic treatments focus on the downstream changes in dopamine, but they do fail to address the primary abnormality. I don't know why that word's so hard to say. So antipsychotics target postsynaptic dopamine signaling, while dopamine dysfunction is largely due to aberrant presynaptic regulation. So the antipsychotics aren't treating the cause of the dysfunction, they're only treating a symptom. And then presynaptic dopamine also likely lies downstream of other neurotransmitters and circuit changes. So it's actually been proposed that rather than focusing on dopamine, perhaps we should be focusing on glutamate or GABA that's actually upstream from these dopaminergic dysfunctions. So that's going to be talked about a bit later, and also in some of the papers that you guys are going to possibly present, so they talk about looking upstream from even just the dopamine systems. So because of these questions, the dopamine hypothesis 2.0 didn't really stick around that long, but now we are focused on the dopamine hypothesis 3.0. So this is the new and improved current dopamine hypothesis, and this one states that schizophrenia results from combined genetic and environmental insult resulting in altered neurodevelopment, including subcortical dopaminergic hyperactivity and psychosis. So this hypothesis is really taking into account the genetic and environmental impacts of that two-hit hypothesis we talked about on Wednesday, and kind of integrating that into the dopamine hypothesis 2.0. But then separating out psychosis from just lumping it in with schizophrenia as well. So again, there are other neurotransmitters in schizophrenia that are involved other than just dopamine. So atypical antipsychosis are those having activity at sites other than the D2 receptor. So clozapine, risperidone, and quintiapine are those antipsychotics. You don't really need to remember those ones specifically, but just know that atypical antipsychotics act at different sites other than D2. Common binding sites actually include serotonin 2A receptor, And atypical antipsychotics often have reduced extrapyramidal side effects and are more effective against the negative symptoms, whereas the typical antipsychotics are more effective against the positive symptoms. Aripiprazole is a partial agonist to the D2 receptor. So what it does is it increases signaling at the D2 receptor symptom with low dopaminergic tone. So this is in the prefrontal cortex. And then it reduces the signaling at the D2 receptor symptom with high dopaminergic tone, so in the mesolimbic system. So it kind of does the opposite effect based on where it's acting. So partial agonist acts as an agonist, but also elicits a less than complete activation of the target receptor. So it can increase signaling, D2 signaling in the absence of dopamine, but it also reduces the signaling in the presence of high dopamine levels. So in other words, if there's no dopamine around, it's as a partial agonist, it's going to act as dopamine. But if there's too much dopamine around, then it's going to act more as an antagonist. So the glutamate hypothesis really focuses on NMDA receptors in schizophrenia. So agonists such as ketamine and PCP of the NMDA receptor produces positive symptoms as well as negative symptoms. So you'll see hallucinations and delusions, as well as some apathy and anhedonia. And then you also see some frontal cognitive deficits. So really using the antagonists such as ketamine and PCP can actually be used in animal models to kind of induce schizophrenic-like symptoms, where we can then test treatments. Haloperidol is a first-generation antipsychotic, and it increases the NMDA receptor density and extracellular glutamate concentrations. So in other words, it kind of brings more NMDA receptors to the synaptic cleft so that there's more chance of these agonists to work. So we do see some elevated glutamate levels that are reported in patients taking antipsychotics. So that would kind of support that there's increased number of NMDA receptors because there's now more glutamate when taking these antipsychotics. So there are multiple genetic risks that are associated with excitatory synaptic function. Again, we kind of talked about that. We had some nice graphs in the genetic risk video. And then oral administration of glycine, which is an NMDA receptor coagonist, has been reported to improve negative symptoms in patients. So while glutamate antagonists like ketamine and PCP can induce both positive and negative symptoms, using glutamate as an antipsychotic again usually only treats the negative symptoms and not the positive symptoms. So like I said, you can use NMDA in animal studies. So we usually kind of focus on the NMDA receptor 1. Partial depletion has been shown to cause some behavioral abnormalities in mice. So without these receptors, then you don't get as much glutamate into this postsynaptic cell, which will then cause these behavioral abnormalities. So that, again, is kind of similar to those antagonists that block the NMDA receptors, and we see the symptoms. So this is consistent phenotypes to the PCP treatment. So the MDMA receptor 1 mice also respond to typical and atypical antipsychotics. We see that haloperidol, which is a typical antipsychotic treatment, ameliorates hyperactivity. So that's a proxy for positive symptoms in animals. Obviously, it's hard in animal studies with schizophrenia because you can't really ask them if they're seeing or hearing any sort of hallucination or ask them about any delusions. So we kind of have to focus on observable positive symptoms such as hyperactivity. And then clozapine, which is an atypical antipsychotic, treatment for that ameliorates hyperactivity and social deficit. So these are a proxy for negative symptoms. Again, it's showing how NDMA R1 studies, R1 partial depletion studies are a pretty good model for schizophrenia. However, and it's, you can see how two different types of antipsychotics treat the symptoms. However, it is hard to see all of the different symptoms. We can also use animal studies as well as some human patients to do post-mortem evidence. So post-mortem analysis from chronic schizophrenia patients has shown decreased glutamate in the hippocampus and prefrontal cortex. So again, this really relates to what we were talking about with like up here with the glutamate hypothesis, how you need glutamate for proper function, and how blocking the glutamate function using drugs can imitate schizophrenia. So here we're seeing that patients actually have a decrease of glutamate naturally, which makes sense. So we also see an increased activity of glial glutamate transporters, and this has been observed in post-mortem tissues from schizophrenia patients as well. So we talk about how there's glutamate deficits, but there's also some evidence for GABA deficits in schizophrenia as well. So similar to glutamate, GABA is kind of higher in the pathway from dopamine, so that's why we're still talking about it with the dopamine hypothesis, because while we have dopamine deficiencies, then the problems there are likely from something happening higher above it. So with the glutamate and the GABA deficits. So GABA is the primary inhibitory neurotransmitter in the central nervous system. We learned that back in Psych 104, and I'm sure in other classes. So it decreases the likelihood of the postsynaptic cell firing. The GABA receptors are ligand-gated chlorine channels that hyperpolarize the target membrane on activation, so it makes it more negative. So the deficits in glutamic acid decarbolase, or GAD, that's this thing here, have been identified in the prefrontal cortex in schizophrenia patients. So I just want to go over kind of this pathway here, because this is something that we're really going to kind of focus on and is thought to be involved in schizophrenia. So we have GABA goes into the synaptic cell, or synaptic gap, obviously. It can bind to the GABA receptors, and then it's taken back up by the GABA transporters, GAT1, 2, and 3, into an astrocyte. And then in the astrocyte, it's broken down from GABA to glutamate to glutamine. Glutamine transporters then transfer it back into the presynaptic cell, where it's then turned back into glutamate. And then from there, it goes from glutamate to GABA via this GAD, the glutamic acid decarbolase. Okay, so that's its role. So deficits in GAD mean that we don't get GABA, so we don't get the inhibitory acts on the postsynaptic cell. Instead, it stays as glutamate. And we know that glutamate is excitatory. So if glutamate's released into the synaptic cell instead of GABA, then it would be excitatory instead of inhibitory. So that's where we really see some problems going on. So that's kind of the main circuit I want you to focus on. There's the secondary circuit where GABA gets taken in by GAT1 straight into the presynaptic cell and is then recycled via GABA aminotransferase, but you don't need to know that as much. If you want to know it, cool, but really focus on this outer circuit. So in summary, the dopamine hypothesis 3.0 is really a combination of studying not just dopamine, but also other neurotransmitters that are involved kind of up-circuit from it. So we have that dopamine is a neurotransmitter system targeted by antipsychotics. Those antipsychotics have a good effect on the positive symptoms by focusing on the D2 receptors in the mesolimbic system. However, some negative side effects are the extrapyramidal side effects. So remember, those affect the kind of nervous system, the nigrostriatal system. VTA to the NA, so the nucleus accumbens circuit, moderates the sensory salience in psychosis. So when you interrupt this circuit, then that's where you get too much dopamine into the nucleus accumbens, which leads to psychosis. Amphetamines will release dopamine to produce psychosis indistinguishable from schizophrenia, so another way to kind of induce schizophrenic symptoms in animal models. However, dopamine, then the causality of the dopamine changes is still unclear, and that's what's led us to look upstream at these other neurotransmitters. So when looking at serotonin, then atypical antipsychotics act at the serotonin 2A receptor, and these atypical antipsychotics tend to have better effects on the negative symptoms than the positive symptoms. And then looking at glutamate, then MDMA antagonists like PCP and ketamine are great at producing both positive and negative symptoms of schizophrenia in humans and animal models. So a lot of animal models of schizophrenia will focus on this. And then antipsychotics will increase the glutamate levels as well as the NMDAR1 density, which you can see when you look at treating NMDA antagonist-induced schizophrenia when treating it with both atypical and typical antipsychotics. And then finally, looking at GABA, we see that there are deficits in GABA metabolism, which is associated with schizophrenia risk. And we talked about that whole circuit and what's involved there. So that's it for today, and on Monday we start the depression student presentation, so it'll be 2 weeks until the next video is posted.